Hello students, I am Dr. Madhusree Halder, Department of Botany, Sri Rampur Girls College. I think all will be okay. Welcome to the next semester. Here we have to study a lot of molecular biology and today my topic of discussion is transcription in eukaryotic system. First thing we have to know that why RNA or transcription process is the heart of the gene expression, or why this all thing is important for our life. So we have to study briefly the central dogma, that is the production of RNA, that is a ribonucleic acid, from double-stranded DNA molecule via the process transcription with the help of one enzyme known as RNA polymerase. Then from this RNA molecule proteins are being synthesized, and here major three types of RNA are produced from DNA, that are mRNA or messenger RNA, tRNA or transfer RNA, and the third type is rRNA or ribosomal RNA. That's why this transcription process is so much important, because these all types of RNA are important for translation process by which proteins are synthesized. Let's have a glance in the system of both the system, prokaryotic and eukaryotic system of transcription. At the left hand side here is the prokaryotic system, and at the in the right hand side there is eukaryotic system. In prokaryotic system you can see that there is no definite compartment for storage of the genetic material or the nucleic acid, and the genetic material are being naked in the cell. And important thing is that here transcription and translation process are simultaneous. That is, on the one hand transcription is an ongoing process, you can see that. On the other hand, translations are also started. That's why transcription and translation are simultaneous, because there is no definite compartment, all are at the same place. That's why it is possible. And another important thing is prokaryotic mRNA is polycistronic, while in eukaryotic system there is a definite compartment for storage of not only storage, for bearer of the genetic material. And here from DNA a primary transcript is produced by the process of transcription, and after some post-transcriptional modification, namely capping, tailing and splicing, a mature mRNA is produced with a 5' capping and 3' poly A tail, with the addition of this poly A tail. And after that, this mature mRNA moves out to the cytoplasm for translation, and here with the help of ribosome it can produce some protein. So here transcription and translation are not simultaneous process in case of eukaryotic cell, and eukaryotic mRNA is monocistronic in nature. Next thing is RNA polymerase. Here also some difference in two systems. You can see that in case of prokaryotic system, RNA polymerase enzyme is single, but in case of eukaryotic system there are three types of RNA polymerase, namely RNA polymerase 1, RNA polymerase 2 and RNA polymerase 3. Only the names are not different, but their function and the place of their occurrence is also different. In case of RNA pol 2, it is found in nucleolus and the function is to synthesize the 45 is rRNA, while in case of RNA pol 2 it is found in nucleoplasm, and the function is synthesis of hnRNA and mRNA also. But in case of RNA polymerase 3, the occurrence is at the nucleoplasm, and they synthesize tRNA as well as snRNA or small nuclear RNA and some rRNA. Also, another point of difference is there, that is the size of the RNA polymerase. The prokaryotic RNA polymerase is smaller than that of eukaryotic one, because prokaryotic polymerase enzyme, RNA polymerase enzyme, is made up of 2 alpha subunit, 1 beta dash, 1 omega and 1 sigma subunit. But in case of eukaryotic system, suppose it is a Saccharomyces cerevisiae or yeast, the RNA polymerase 2 is made up of 2 large and 10 smaller subunits. Next part is the promoter region. Here is also some difference, because in case of prokaryotic system, you can see that at the left hand side the start site is here, and that is designated by 0, and the upstream site is designated by minus. Here two region are there. This upstream or the minus region is known as promoter region, and here are some consensus sequence or conserved sequence. In case of this E. coli cell, two consensus sequence are -10 box or Pribnow box. The sequence is TA80, that's why it is also called Tata box, and -35 box. But in case of eukaryotic system, upstream to the start site there are Tata box or Goldberg-Hognes box, cat box, GC box, as well as enhancer or silencer. So what is the function of enhancer or silencer? The answer is, the function of enhancer or silencer is to speed up or gear up the rate of transcription or reduce the rate of transcription while needed. And interestingly, these all elements are cis-acting elements, because these all are found at the same side of the start side and at the same side of the same DNA strand. Next thing is initiation of transcription. In case of prokaryotic system, the initiation is very much simpler, because the sigma factor of the RNA polymerase enzyme is very much important for recognition and binding of the binding of the promoter region. And in case of eukaryotic system, there is a lack, or this polymerase enzyme are unable to recognize or specifically unable to self-recognize the promoter region. That's why some transcription factors are needed, and they play important role in recognition to the promoter side and binding to it. And these all transcription factors are known as trans-acting elements, because they interact with the cis-acting element and they are external to the cis-acting element. After that, they facilitate the binding of RNA polymerase to the promoter region and activation of RNA polymerase. The term activation of RNA polymerase is a question mark to you. I will explain it later. And then initiation of the transcription. And another important thing is that here is a pre-initiation complex. So let's see what is the process. Before that, these are the transcription factor. Names are TF2A, TF2B, TF2D, TF2E, TF2H and TF2F. So at the right hand side, these are the function of this transcription factor. So this will also be clear to you in a diagrammatic view. So that is the formation of pre-initiation complex. The process initiates with a double-stranded DNA molecule, and at the Tata region or the promoter region, first of all, the TF2D, it has two subunit, TBP and TAF, that is the Tata side binding protein and Tata binding protein associate factor. They came and bind to that. Next, two transcription factors, TF2B and TF2A, to stabilize the TF2D with the Tata site. Next, another two protein, one is TF2F and mediator protein, along with the RNA pol 2, came to the site of initiation, and they help in binding of RNA pol 2 just ahead of TF2A. Next, another protein, TF2H, that is very much important for activation of RNA polymerase 2, because this enzyme or this transcription factor phosphorylase the C-terminal domain of the RNA polymerase 2 and activate it. Next, another enzyme, TF2E, that actually helps in unwinding of the double-stranded DNA and to initiate the proper initiation and elongation phase. Next, at the start of the elongation phase, the entire complex remain unstable, because you know that while rail engine started, and at the car shed, only the engine will move many times, or starts and move many times to check the whole system. The same thing also happened here. The pre-initiation complex along with the RNA polymerase wants to check the whole system is okay or not. That's why RNA pol slides downstream to check the system, and a short sequence of RNA will formed, and then it stops and the RNA pol back to the position of the start point, and that's why it is called abortive transcription or abortive initiation. Only after that a stable DNA RNA polymerase 2 complex is formed and elongation continues. At active phase of elongation, the enzyme complex is made up of RNA pol 2, 40 base pair of DNA and 18 base pair of RNA. Another important thing is that at RNA polymerase enzyme, there are two pores at the upper side and the bottom side. One is called lid, while another one is called pore. You can see in the diagram that these two are the pore, lid and pore. At the bottom side, all the RNTPs go inside and form the RNA chain, and from the outsider lead, the growing RNA chain comes out. But another important thing is that what will happen if a nucleosome complex come in the way of the RNA pol 2 during elongation? It will stop? No, the answer is no. Another protein, chromosome remodeling complex, comes and just slides the histone octamer core along the DNA to make the way to RNA pol 2, and the process will continue. Okay. Next thing is the termination. Important thing. It is unlike to that of prokaryotic system. That is, in case of prokaryotic system, either that is row dependent or row independent. But in case of eukaryotic system, there is a specific endonuclease enzyme that cuts the RNA strand, followed by a polyadenylation at the 3' end of RNA and transcription ends. Thank you. I think this video is helpful for the students, so carry on.